So good morning all of you. We did our portion on different types of diseases in the last class. So just developing on that concept of health, we are going to talk about immunity. And I guess the world we are heading into, again, it is very important that we make sense of the terms which we keep hearing all around us. So we'll start with this question, UPSC previous year's question. So please solve the question first. So the question says, which one of the following statements is not correct? So not is the important point here. The answers I am getting are A and B. Hepatitis B virus is transmitted much like HIV. So the transmission of HIV is, out of the many ways, it is through blood and body fluid transmission. And that is the same for hepatitis B. So this is correct. Second statement is hepatitis B, unlike hepatitis C, does not have a vaccine. So we studied last time that out of the different hepatitis viruses, hepatitis C is the one which does not have a vaccine. So this statement is incorrect. It should be the other way around. Hepatitis B does have a vaccine, but C does not have a vaccine. So second statement is incorrect. Then third one, globally the number of people infected by hepatitis B and C are several times more than those infected by HIV. This is absolutely correct, and this fact UPSC had asked earlier also, that hepatitis is more infectious than HIV. This was the statement which was there. So this is also correct. Some of those infected with hepatitis B and C viruses do not show the symptoms for many years. Yes, this is very much possible, both with HIV and with hepatitis, that you can have the virus in your body, but you might not have any symptoms. For HIV, it is such that for decades, for 10 years, for 20 years, there is a possibility that you have the virus. That means you are HIV positive, but you do not have the disease. You do not have AIDS, but you have HIV virus in your body. Similar is the story with hepatitis viruses. They can stay in your body without giving you any symptom, any disease. So this is also correct. Now answering this question should have been easy if you just focused on this aspect of vaccine. It's the easiest of all statements. So even if you are getting confused in the last two, C and D, right, maybe you have not read about it or you're not aware, then in that case also this should easily come. This is an objective fact, this you should know and this you would know. So this question, they've asked you to pick out an incorrect statement and that is B. B is the right answer. And I guess most of you did get that, but for those of you who did not get that, and there are some other options which you are finding them to be new, so I hope you have already noted them down. Answer is B. Next point is talking about the concept of immunity. So, can you first tell me what is your understanding of immunity? According to you, what is immunity? Let's read a few answers of yours. So tell me, what do you think is immunity? Everywhere around the world, there is this big discussion on immunity, how to improve immunity, what needs to be done, what exactly do you think is immunity? What is your understanding of immunity? Tell me that. Ability to fight disease, according to Keshav. Okay. So you see this picture also. There is a man, he's holding a shield and there are some microorganisms outside the shield which are obviously coming to attack him. So this shield is the immunity. Potential to fight diseases, ability to resist infection, antivirus. Not just antivirus, all against all pathogens. Capacity of body to fight diseases. Any shield developed by the body. Capacity to resist an antigen. Achha. Ability of a body to fight diseases and keep the body healthy. Okay, good. So you understand the basic idea. Basic idea is how the body will be protecting itself. So that is the body's basic self-defense. Body's basic self-defense to protect itself against any infections which are going to be taking place. That self-defense is immunity. So any invasion, any attack from outside which is going to take place, the body is going to have a mechanism to combat that, and that is what is immunity, that is what is the immune system is about. Alright, so let's write this down. Please write this down. Immunity is the balanced state of an organism having adequate biological defenses to fight infection, disease, and other unwanted biological invasions. It is the balanced state of an organism having adequate biological defenses to fight diseases, infections, and other unwanted biological invasions. Now we'll talk about the different types of immunities which are there. Okay, the different types of immunity or the different processes which the body is using, immune processes. Let's talk about that. Okay, so you're telling me there is some sound disturbance. We'll just get it checked. Okay, so we have informed the technical team about it. So I guess they'll look into whatever is the issue of sound. We are categorizing immunity under two sections. So this is innate immunity and acquired adaptive immunity. Innate immunity and acquired adaptive immunity. So what are these two terms meaning? First of all, we'll talk about the innate immunity. Okay. So innate immunity is something which is already present with the body, something which you are inherently born with and will be active, will be available even when there is no attack, no problem. Right? So we are talking about how immunity is the attack against the different kinds of diseases which are coming. So maybe bacteria are coming to cause the disease, virus is coming to cause the disease, fungus is coming to cause the disease, right? So one kind of an immune response in the body which we call the acquired of okay, let me just pause and let's see what we can do about the sound. Okay, so I guess we do not have a unanimous pitch on if there is a sound issue or not. But nevertheless, I'm going to change my mic and hopefully all of you will not be facing any problem. Hence, let's see, let's try that out. I am definitely going to put my hand near the transmitter, Priyanshu, and I'm going to wave it and I'm going to move it also. That's how I teach and that's how I talk. So sorry, that I will not be able to deal with. But yeah, I can definitely change my mic to see if we have a better situation. Alright, so hopefully we will not face a problem now. So we are talking about types of immunity, and in this I am talking about two types. One is innate immunity and one is the acquired immunity. Immunity क्या है? Basically it is the self defense. The self defense with which the body will protect itself against any kind of attack from unwanted agents which is going to take place. Now there can be a form of protection which is already in place, and there can be another form of protection, another response which takes place after the attack, right? Like think of your border, okay, think of the international borders. So there are some forces which are always there. Some forces, okay, they will always be there in the border. Be there an attack, be there no attack, but they are always there. But when there is an actual attack, when there is an enemy attack, then those forces alone are not sufficient. From all over the country, more forces are going to be called out because a targeted high intensity response is needed to combat the enemy. This is the same concept. Innate immunity is what is already present, always present. There might be an enemy, there might not be an enemy, but things in innate immunity, you naturally have them and they are always there. Acquired or adaptive immunity is what happens once you are having an attack, right? You have a bacterial infection, you have a viral infection. So after that, whatever body's response is, that becomes acquired or adaptive response. What was already present, that is the innate response. Okay, we'll take examples in both these categories, then it will become more clear. So innate response mein we can have something like the skin. The skin, and the skin being intact, it is supposed to protect you against any microorganisms going inside. That's why when you have any kind of an injury or a cut on your skin, it is said that you must make sure that you use some antiseptic solution on it. Why we are using antiseptic is because we do not want any microorganisms to enter, because now the skin is discontinuous. Okay, so naturally the skin being there is the first layer of barrier protection which we provide, and this is innate immunity, because the skin will not come in or not be formed once bacteria come and attack your body. That's not going to be the case. It is there irrespective of any attack. Okay, then we have other such things also which are naturally present, right? Before we go into this, there are a few characteristics of innate immunity which you should know. One is the fact that because it is inherently present, it is non-specific. Non-specific as in, like your skin is there. The skin is not going to specifically stop any one kind of microorganism. Okay. The skin will not say, acha, bacteria, you cannot go inside. Oh, virus, you please go inside. It's not like that. It's not a selective reaction. It is there. The barrier the skin is providing, that is for all kinds of unwanted things. Right? So, the response here is non-specific in nature. It is not specific or not different for bacterial infection, viral infection, infection of amoeba, anything like that. Okay. So it is non-specific and it is inherently present. So this is the first line of defense in the body. Okay. Let's write down innate immunity, then we will see more examples. So it will become more clear, and after that we will go to acquired or adaptive immunity. Please write. innate immunity is the non-specific response which is activated immediately on exposure to a pathogen or an antigen. Just write, cut both these terms because we still have to study what is an antigen. So write down on exposure to any unwanted agent. We'll write the blanket term, exposure to any unwanted agent. It forms the first line of defense. In the body. It forms the first line of defense in the body. Now let's see the different categories or the different examples, as we can call them. Please write this down. It is of three types. First one, physical barriers. In this we have the skin and mucous membrane. So like the skin is the outermost layer of your body, mucous membrane is the lining which the organs have. The different organs, they have their own lining, own barrier, and that is your mucous membrane. The skin on the outside and mucous membrane on the different organs. Second category is chemical barriers. In chemical barriers, we have certain fluids in the body which are supposed to have antimicrobial action. So any kind of microorganisms which come, they can be naturally disintegrated or removed from the chemicals in saliva, in tears, and the HCl in stomach. See, tears in human beings, they are also supposed to be catering to a psychological part, a psychological role in terms of what you can say, a psychological response is going to come out from your tears. But along with that, the main function is that of providing a chemical barrier, or you can say protecting the eye, cleaning the eye. In animals other than human beings, the emotional component is not as such related to the coming out of tears. So tears in those animals, the only job of tears is to keep the eyes clean, to keep it free of infection. And if you think of tears also, tears would be there always. You have the property of having tears and of producing tears, and they can immediately be triggered whenever there are some unwanted things. Okay. Third one is the presence of some cells in the body which are readily going around to attack unwanted things, and these are neutrophils and monocytes. They are both white blood cells. Neutrophils and monocytes are white blood cells which are a part of the innate non-specific immunity, and these are cells which are already present in the body doing this job of forming the first line of defense. Is herd immunity also unacquired immunity? So if you want all your questions to be addressed, the first thing is ask them in the doubt section. It's not practically possible for me to take questions from 300 students on the response portal, right? And to answer your question, we will be discussing herd immunity also today, and we will also be discussing vaccine today. So hold your breath, picture abhi baki hai. So let's go about things one step at a time. Let's write about adaptive or acquired immunity. So just like innate immunity we said is non-specific, adaptive or acquired immunity will be specific. When I say specific, that means bacteria के लिए different kind of response, virus के लिए different kind of response, amoeba के लिए different kinds of response. Highly specific, and because of this specificity, it takes more time. So first the innate immunity is activated, and then the acquired and adaptive immunity takes time, right? Anything which you have to prepare through customization in the world, right? So that customization takes time. So adaptive acquired immunity is the customized specific response, so it takes time. So it's a little slower than innate immunity, which is already in place, right? But because it is customized, it would be more effective, right? And we'll see what are its different components. Just write down first. It is the specific immune response. It is the specific immune response developed against pathogens. It is the specific immune response developed against pathogens on exposure to unwanted agents. On exposure to unwanted agents. It is slower than the innate immunity. It is slower than the innate immunity. Okay, so
What about cytokine barriers? So cytokine response is a part of your acquired adaptive response only, and we are going to see different mechanisms of adaptive response now. The first one which we are going to talk about is antigen antibody, and cytokine attack is something similar to that response, but yes, it is a part of adaptive acquired immunity. Okay. Okay. आगे बढ़ते हुए next we are going to do the mechanisms in your acquired adaptive immunity, and the first one is going to be antigen antibody. Now these are terms which we have heard so very often in the last few years. I think they are not new terms for anybody. We talk about antigen test, antibody test, it's always there. So it's important that we first understand what these two words mean. What is the technical aspect of being an antigen or of being an antibody? Okay, so antigen is anything in the body which can trigger an immune response. Now think of this in a way that we said that our body will be having some responses against the unwanted agents. Now this term unwanted agents is important. That means against everything the body will not be giving a response. Think about how many things are going into your body. You're eating food, you're drinking water. If your body starts responding and attacking everything, that means you'll never be able to consume anything. Okay, so not all things are bad, not all things are unwanted, but those things which are unwanted, those things which the body thinks are damaging, only against them the body will have an immune response. Okay, so all those things, now they can be different organisms like bacteria, viruses, amoeba, okay, or other things like pollen grains can be perceived negative, or cells from other organisms, like a blood transfusion we are doing, blood from other people are coming inside your body, or organ transplant we are doing, organs from other parts are coming. So all these things which the body thinks are foreign, all these things which the body perceives as unwanted and damaging, against them there is going to be a response. And that is the definition of antigen. Anything against which the body gives an immune response, that is an antigen. Okay, so whatever stimulates your immune system is not stimulated by everything. There are specific things on who's coming into the body, an immune response will be triggered, and whatever triggers that immune response, that qualifies to be called as an antigen. This is the basic definition. We can develop and make this as complicated as that in terms of concept of antigens and their structure, and a lot of things are there, but basic idea यही है. ठीक है, बेसिक आइडिया, बेसिक डेफिनेशन यही है एंटीजन की, कि कोई भी ऐसी चीज, एनीथिंग विच विल ट्रिगर योर इम्यून सिस्टम, which will trigger the body to start off and give an immune response, that would be an antigen. वो चीज अब कुछ भी हो सकती है. आपने कोई पॉइजनस प्रोडक्ट पी लिया, बॉडी को पता है हार्मफुल है, अगेंस्ट दैट देयर विल बी एन इम्यून रिस्पॉन्स. ओके, आपने कोई अनवांटेड फूड आइटम कंज्यूम किया, उसमें माइक्रो ऑर्गेनिजम्स थे which were damaging, that would be an antigen, right? So it can be anything, antigen can be anything, not just pathogens but other things can also be included in it. But main idea यही है कि जो भी चीज whatever triggers an immune response, that is an antigen. Okay, write this one statement. An antigen is any agent which triggers an immune response. Any agent which triggers an immune response. Okay. Now what is this immune response, or what is the response to an antigen? The response to an antigen is an antibody. When your body recognizes something as antigen, कि अच्छा ये this is an harmful thing which is coming in, so the immune system is triggered, and as a response what is going to happen is an antibody is going to be produced, and this antibody is a protein. Protein produced in response. So the body has recognized antigen. Now I told you in acquired adaptive immunity there is a specific reaction, there is a specific response which is there. What is this specific response? It is the production of antibodies, and antibodies always will be proteins. Antigen कुछ भी हो सकता है. Antigen can be a chemical, it can be a cell, in that cell also it can be a protein, it can be a polysaccharide, it can be a fat layer, anything can act as an antigen. लेकिन जो antibody होगा, it is always going to be a protein. Okay, it is always going to be a protein which is produced in response to an antigen. Now specific का point क्या है इसमें? Antibody in every case will be different. If there is a bacteria, the antibody will be of one kind. If there is a virus, the antibody will be of another kind. And under that also, different kinds of bacteria would give us different kinds of antibodies. That is the specificity. कि हर चीज के लिए अलग एंटीबॉडी बन रहा है बॉडी में. ओके, डिपेंडिंग ऑन वॉट एवर इज अटैकिंग, दैट एंटीबॉडी will be produced. All right, just write this one line. Antibody is the protein produced. Antibody is the protein produced as a response to the antigens. As a response to the antigens. ठीक है? Now just look at this picture, this cartoon which is there. These are the cells of your immune system. Now these bacteria, they have come into the body. So these cells have recognized this bacteria. First thing to trigger a response would mean कि आपको पहले तो ये recognize करना है कि what has come. So they have recognized the bacteria, and as soon as they have recognized, what they are going to do is they are going to start producing specific antibodies to attack these bacteria. Okay, it's basically like a lock and key kind of an arrangement, right? Specificity उसी से आती है, that they make antibodies compatible to the structure of these antigens so that they will be fixed to those antigens and the antigen will be destroyed. Basically antibody का काम है इसको destroy करना. Now these Y-shaped structures which you can see, these are the antibodies. These antibodies will go and attack the bacteria and the bacteria will be destroyed. That's what their job is. So this one says I am hit and it's actually warning the others to please go on without them. Okay. So this is antigen antibody reaction. Very important basis for you to understand so that you can understand the vaccination topic which we are going to do after this. Okay. When you asked me about cytokines, let me tell you about that a little bit. Not exactly on this idea, but mechanism concept is same. So with COVID, I know cytokines have been mentioned in many places. So what is cytokines is that, suppose this is a cell of your body and this is under attack, say by a virus. So viruses as such we know they go into the cell and attack, right? So this is a cell which is under attack, the virus has come. Now what happens is that this cell has a potential of producing special agents called cytokines. What is the role of these cytokines? They are supposed to be protective to other cells. It produces cytokines to protect surrounding cells. See, immunity in the body is a complex process and many different types of reactions and mechanisms simultaneously are going on in the body. Okay. For our understanding, for the technical sense of it, we have differentiated them and we are talking about them separately. But body में ऐसा नहीं है कि either antibodies will be produced or cytokines will be produced or the T cells will be doing their job, we'll see what they do. So it is kind of an integrated approach which the body is using. This is one of those mechanisms that a cell which is already damaged. So if the cell is like कि भाई मैं तो गया तुम लोग बच जाओ. That is the idea. So this cell produces cytokines so that the surrounding cells, the cells around it which are not being attacked, they are protected. That is what is the cytokine mechanism. गायब हो गया? Okay. Okay, that is what is the cytokine mechanism. Now, how you know about cytokines is that in COVID, there is this idea of cytokine storm which is taking place. Now, what is this cytokine storm? This is an I'm trying to keep it very non-technical in the most simple terms so that you can understand and make sense of this. So, ये जो cytokine है, normal mechanism in which infected cells try and protect the other cells, sometimes there is an overproduction of this. जितना जरूरत है उससे ज्यादा cytokines when they are produced in the body, that causes a condition called cytokine storm. And ये अब damaging है. Something which is supposed to be a protective mechanism, that can be damaging. Okay. And with this kind of a situation which people are experiencing with COVID infection, so there is an outburst of excess amount of cytokines, जिसकी वजह से, because of excess of these cytokines, there is excessive fever which a person is having. There are even cases of excess swelling in the body and even death because of the sudden rise of cytokines. Okay, so this is a condition which is associated, right? I hope you understand the concept. so that we can talk about the next idea. Okay, Mayank has asked, you said that self-medication can lead to antibacterial resistance. Is the self-medication can affect non-infectious diseases like disorder or anxiety? Okay, not exactly on the theme which we are discussing about, but yes, all kinds of medications, Mayank, be it a simple medication like an anti-anxiety medication or antihypertensive medication, all medicines have a potential of becoming resistant. Of course, the mechanism or the kind of cells which are becoming resistant, they are different, right? But if you're talking about medicines becoming ineffective in non-communicable disease, yes, the answer is yes, that can definitely happen. Okay, but because your infectious diseases, they are in an acute form, and if they are infective, there is chances of a person dying also before it is identified and diagnosis. So the severity and the scale at which antibacterial infections are and their resistance is discussed, that is a little wider. But all medications can become resistant and ineffective on a person. Right? That is why there is always this discussion of having a consulted approach towards medication, making sure you are taking the appropriate dose and the appropriate amount. Which organ gland produces antibodies? So, Shubham, white blood cells produce antibodies. We will talk about how that is working in a while from now. Can we say antibiotics are foreign external antibodies? Absolutely not. Lakshya has asked this question. So, antibiotics or antibodies को बिल्कुल confuse नहीं करना है. There is no correlation between them other than the fact that these words are confusing for you. Please remember this. Antibiotics are chemical agents to attack, to destroy the bacteria, only for bacteria, right? They are drugs, they are medicines, that's it, that's what their role is. This is not a part of body's inherent immune response. Okay, so isko separate rakhna hai, even when the name seems the same. This has nothing to do with antibody. Is a very advanced type of a reaction of the body, and they will always be proteins. तो वो body में ही produce होती है, वो proteins ही होती है. Antibiotic is just something which we are doing and giving from outside for bacteria specifically. ठीक है? Please tell, nutrition and minerals also can be unwanted invasion in the body which are taken from outside, or they are a part of immunity. So nutrients as such, there is no damaging effect or antigenic effect of nutrients. There can be chemicals, some form of chemicals which can be harmful, but the minerals and nutrients which are there, we are supposed to be taking them, the body needs them, so they are not unwanted invasions. Why no antibody for lactobacillus found in curd? That is the point, Vibhu. That is what we are saying, discussing here, कि हर चीज के against response नहीं है. Everything in the body is not going to trigger an immune response because everything is not damaging. Lactobacillus, or leave lactobacillus. Lactobacillus to chalo we are taking from outside. Otherwise also there are hundreds of bacteria and fungus which are naturally living in your body, and some of them are very helpful. Like we have bacteria in our intestine which are supposed to do the job of producing different vitamins. Okay, so they are helpful, healthy bacteria. So why would the body want to attack them? Not everything that goes into your body, please understand this, इसीलिए ये concept है. हर चीज antigen नहीं है. Everything that you are taking and that you are consuming, that is there, they are not supposed to be damaging. So इसीलिए हर चीज के against antibody will not be produced. only those certain things which can trigger an immune response, that means which are perceived as damaging, they are the ones who are going to be specifically picked up and targeted. Not everything. If everything, just think about this, common sense ये लगाओ कि अगर everything in your body which is coming, if the body is attacking all of those things, then you will never be able to have any form of nutrition. आप खाना खा ही नहीं सकते. Because the moment you eat something, your body will give an immune response. You will have antibody production, you will have high fever and all those other responses which are associated with it. So that's not the case, right? You are supposed to take in some things are not damaging to you, so they would not be antigen. Antigen वो ही चीज है जो body perceive कर रहा है कि this is damaging. So whatever triggers the immune response, that is what we are going to call antigen, and everything is not an antigen. That cannot be the case. Deepshika has asked, are cytokines only produced in the viral attack or any pathogen? No. Cytokines का ऐसा कोई specific relationship नहीं है. Virus, I just gave you the example of virus because it has become more famous with the recent COVID-19 infection. But otherwise it's a normal response of the body. It can happen in many cases. Allergic reactions may it is very predominantly something which is coming out. Okay, Deepshika? Yeah, antigen is the cause and antibody is the consequence. Correct. एक और mechanism, immune mechanism. Let's talk about one more mechanism in which there is no antigen, antibody or response as such, but there is a process in which cells can be destroyed directly. Cells can be destroyed directly through a process called phagocytosis. So this is another form of body's response. What happens? This is a cell. The cell sees a virus. कि अच्छा एक virus आ गया. Now what this cell will do is, this is going to try and cover this. We have something like an invagination, like this cavity you can see, this invagination is formed and the virus is engulfed. It engulfs the virus with its plasma membrane, finally bringing it inside its structure, into its structure and then destroying it. Once it is destroyed, you know the cell has digestive enzymes, lysosome if you remember. So they will use the enzyme, they will destroy this viral cell, it will be stored, they have called it phagosome, this is nothing, vacuole mein, whatever the products are found after destruction that will be stored in the vacuole and the cell will be destroyed. So this mechanism is like killing the foreign unwanted body. Okay, so एक mechanism of attack which we have done is production of antibodies. You have an antigen, you recognize that and you produce antibodies against it, one response. Second kind of response which the body can have is actually taking the cell, bringing it into its structure and then destroying it. Natural killing mechanism. Okay, that is what is called phagocytosis. So write this one line further. Phagocytosis is a process. Phagocytosis is a process by which a cell uses its plasma membrane. The cell uses its plasma membrane to engulf a large particle, and then digest it. Engulf a large particle and then digest it. Praveen has asked, how can the cells know what is wanted, what is unwanted? So that recognition property is there with some of these cells, and it is based on the structure. There are certain structures which they identify, which the body knows are damaging. That identification process of the cells, the cells of the immune system, that is based on certain structural components or certain structures which they are programmed to identify. तो वो identify उस structure को करेंगे and that will be called the antigen. Now how do they know that? Do they have a directory? Do they have some form of book or data or Google to tell them that? Right? That kind of a thought process I know you are thinking with, but we do not really have the possibility of going into that concept. I can just tell you this thing, that there are some things in the structure of an entity which will determine if they would be an antigen or if they would not be an antigen. Okay. Let's talk a little bit about this response further in terms of the cells which are involved. So in your body we have white blood cells called lymphocytes. We have white blood cells called lymphocytes in your body. Now all the blood cells are produced in the bone marrow. Can you tell me where the bone marrow is found? If you have heard this term, where is the bone marrow found? Bone marrow is where the production of cells is going to take place. The white blood cells are white blood cells, red blood cells also, blood cells, they are produced in the bone marrow. Out of these white blood cells, the one cell which I am talking about is the lymphocyte. These lymphocytes are of two types. One is the B cells, the other one is the T cells. In the knee, inside the bone, inside the spinal cord, muscles, thigh bone. Okay, so basically this is the inside the bones of course, but in the your long bones. That means your arms and feet. They are the long bones of the body. That's where bone marrow is. It is the soft tissue inside the bone. Those people who are non-vegetarians, they will identify a little bit more with this concept, because the bones inside are going to have a soft, chewy, grey colored matter normally, which is there. And for the standard understanding of bone marrow, we have to go with the idea that it is found in the long bones of the body, and they are places where blood cells are formed. Now, B cells and T cells, both of them, like all blood cells, they are formed in the bone marrow. Lekin, for T cells, there is a slight exception, that after being produced, they go to another organ called thymus. There is a gland called thymus gland, and in this gland maturation of T cells takes place. So this is a small gland in your neck, Okay, and the job of this gland is to mature the lymphocytes, and those lymphocytes which go to the thymus gland for maturation, they are the T cells. They would both be produced in the bone marrow, B cells, T cells, but T cells का ये exception है that they go to a gland called thymus gland for maturation. Okay. Now, B cells, their job is to produce antibodies. The antibody production which we took into account, which we just now studied, who or which cells are producing the antibodies? These cells are B cells or B lymphocytes, we can call them. Okay. And the second mechanism which we studied, that was the phagocytosis or naturally killing the cells, that would be done by the T cells. Now they are both going to be working together, right? It's just the action which they are performing that is different. So the response which B cells have, we can call this antibody mediated immune response, and the response of T cells, because it directly involves the cell and then destroying of the cell, this would be cell mediated immune response. Their mechanism of action is the same, is different, but the thing which they are going to do is to destroy unwanted things in the body, that is the same. Just how they are destroying, that is different. Shashank has asked, what is the lymphatic system? So we have the main circulation system in the body, that is the system of blood. Okay. Along with this, there is another system of transportation of circulation in the body, which is the lymphatic system. So there is a liquid called lymph, and there are these lymphatic cells or lymphocytes, which are the basic component of that lymph. Okay. And without going into any details of the lymphatic system, what I can tell you is that because of presence of lymphocytes, they are essential for the immune system. What you just need to know is that they are the transport system, the circulatory system in the body. Lymph, there is a liquid and there are some cells. Lymph will not have red blood cells. That is one special. They can have white blood cells, lymphocytes they will have, but they do not have red blood cells. Okay, I hope till here it is clear. I am not dictating anything here. You just have to write this down, that copy it as such. Lymphocytes. Lymphocytes are two types, B cells and T cells. B cells produce antibodies, so they give us the antibody mediated immune response, and T cells, they are directly going to be destroying the cells. They are also called natural killer cells. And their response would be cell mediated immune response. All the blood cells are formed and are produced in the bone marrow. T cells have a distinction of going and maturing in the special gland called thymus gland. Okay. See, in this portion, understanding all the steps, understanding all the terms is very important, because this is what is going to build up or take us to the vaccine portion. Alright. So we are going to now see the mechanism of how this whole system of antibody production takes place. Okay, it can be a little bit new for most of you, and based on that idea we are also going to see what is the idea behind vaccination, right? But because that is a long topic, so I will take a break here and then we'll continue. Okay, Sai has asked me, can you use the dark background on the board please? Okay, I'll do that. Okay, let's take a break, because the topic which we will be doing after this, that's going to take a little bit longer time. So just give me 5 minutes. So welcome back. Let's talk about B cells and how this exact mechanism of antibody production is taking place. अच्छा, क्या बोला था आपने? डार्क बैकग्राउंड. So we have B cells present in the body, and there is an antigen. Now this antigen can be anything. Antigen can be a bacteria, antigen can be a virus, antigen can be some poisonous substance which you have consumed, antigen can be some foreign cells. Anything which we have said can trigger the immune response, that is the antigen. ठीक है? So we have an antigen coming into the body. This antigen has come for the first time. So this is the first exposure, we can say, to this antigen. Now, for B cells to start producing antibodies against this antigen, it has to first recognize it. There are certain specific structural components which I have told you, because of which something is an antigen, and B cells recognize that. कि अच्छा, this is the antigen, then it will study it, then it will say कि okay, now I have understood the structure of the antigen, so now I am going to produce the antibodies. So B cells produce antibodies which is specific to this antigen. Okay. Now at this point of time, because the B cells have encountered that particular antigen for the first time and it has to produce antibodies, so this is going to be a slow and low intensity response. Antibodies are produced, but it is a slow and low intensity response. Once these antibodies are produced, what they will do, what is their job? Their job is to destroy the antigen. That is the basic funda which we have studied. Antibodies specifically produced for one particular type of antigen will do the task of destroying that antigen. In this case, because this is the first time the B cells have seen this antigen, so it takes some time, so it is a slow and low intensity response. But one very special feature is there with the B cells, and that is that along with production of antibodies, they also have the property of remembering this whole exercise, and we call this immunological memory. that means that B cells will now remember this antigen and will also remember the response they gave to this antigen, that is the antibody. Okay, they will have memory being created, stored, and which will remember the antigen and how to tackle it, that is how to and which antibody is to be produced. Now how will this be used? This will be relevant when this same antigen comes into the body a second time. So second exposure to the same antigen. This very antigen which came earlier, when this comes again, what happens now? Now the B cells are in a position to recognize them, recognize this antigen because they have already tackled it once and there is immune memory which is present. So this immune memory will recognize the antigen and B cells will now produce antibodies again. But this time, because things are already there in memory, the response which will be there will be a high intensity fast response. So immediately so many antibodies will be produced because that whole time which was needed to recognize and then act and other such things, that is not needed now. So a very high intensity response will be produced. Now how is it practically relevant, this basic concept? I'll give you an example, try and understand this. So what happens is, think of a disease like chicken pox, okay, a disease like chicken pox. In that what happens, that normally it is said for normal people, most of the people, chicken pox happens only once, right? So why is that? That is because of this immune memory which is there. Let's take the example. You have chickenpox virus coming into the body. Now this chickenpox virus is the antigen for your body. Okay, it is an antigen. When the virus will come, it will trigger the immune system. B cells will recognize this antigen and they will produce the antibodies. But because you have encountered this virus for the first time, there is this time which is going to be there. So you will suffer from the disease and there is a slow and low intensity response, because of which after 15, 20 days, 30 days, whatever is the time, variability for different people, your disease will be over because antibodies have been produced and that virus which is your antigen, that has been destroyed. Okay. Now, but what we have in place is an immune memory, and for chicken pox this immune memory can exist for a lifetime, right? So what is the significance of this? After this one infection which a person has had, there is a subsequent infection with the same virus. Chicken pox virus comes in contact with the person again. So second exposure to the same antigen, to the same virus. Now, the moment this antigen comes, there is an immune memory which is in place, which is going to immediately recognize the antigen ki acha tum to pehle bhi aaye the, right? And once it has been recognized, what it will do is it will be producing antibodies, and it will be producing so many antibodies that before the virus can do any damage, the virus is destroyed, eliminated, because of the high intensity fast response which will be given due to the immune memory. And with that fast response, the antigen is destroyed and the person does not get the disease even when he is coming in contact with the virus again. Okay, is this clear? You have an immune memory. ठीक है, अभी आगे इसमें मत जाओ. Don't ask me question about other diseases. Just wait on. Everything will be clear. Please stop asking any questions on this कि ऐसा क्यों हुआ या उस disease में ऐसा क्यों होता है. I will clarify everything, but you have to give me 10 more minutes for that. Okay, so just hold on and listen. Make sure whatever I am saying उतना कम से कम clear है. The idea of chicken pox, if that is clear, just hang in there. We'll talk about other things. Concept क्या है? I am trying to go slow because I understand this can be slightly confusing. Please pay attention. We have an antigen. In this case, we are talking about chicken pox virus. It comes into the body. B cells recognize, produce antibodies. Slow and low intensity response. But immune memory is created. So any time after this, immune memory for chicken pox can exist for a lifetime. So any time after this, when the body comes in contact with the virus, the immune memory would be triggered, a lot of antibodies would be produced and you will not get the disease. Okay? Now, why does this type of an immunity not form for other diseases? Why are we saying this chicken pox happens only one, but most of the diseases we say they keep happening again and again, right? The reason for that is that this immune memory, how long does it stay in the body, that is variable. For different diseases, we have a different time duration for which immune memory stays, right? You can say this idea in the simple term. People say that we are immune to a particular disease, or we have immunity which has naturally come into our body with the infection. So people who have had chicken pox, they become naturally immune, right, without taking any vaccination, any other treatment, anything, but they naturally become protected to the disease. But that is not the case for all diseases. You can see examples of diseases like dengue, malaria, the tropical vector-borne diseases. They keep happening every year to the same people. Why? Because natural immunity or the existence of immune memory is not long enough. The duration of this immune memory can be different. For some diseases it can be 6 months, for some diseases it can be 1 year, for some it can be 5 years. So based on that, after the period of immune memory is over, the period of immunity is over, people can get that disease again. Okay, I'll give you the example of COVID-19 here. Okay. Wait for me, I'm again saying this, I know you might have a lot of questions, just wait for me to finish this whole idea of vaccines, then I'll give you a chance to ask your questions. Just wait. Now, for COVID-19, I was telling you, we have cases of reinfection normally coming up. Okay, people who have had COVID once, they can have COVID again. But there is a duration. It is 2 months, 3 months, 4 months like that, but there is some duration in which they will not be infected again. That means the immunity or the immunological memory is existing for 2 to 3 months or 2 to 4 months standard time. After that, this immune memory is destroyed, it is removed and the person can get the disease again. Right? Now, what is the basis or how is it determined that how long the immunity or this immune memory will stay? That will be variable for different pathogens. It is based on the structure of the pathogen, and as that structure of the pathogen changes, this immune memory can also change. So how long you will stay protected against a disease, that depends on that pathogen. For some, the immunity is more, like chicken pox virus, lifelong immune memory. For others like COVID-19, a few months. Right? Now, we can use this idea to create something which we call the vaccine. Vaccine mein, what we are doing is we are going to exploit this idea of immune memory and develop use the body's mechanism to provide protection. How are we going to do that? That is the next question. Let's see. All right. So, we have a particular pathogen for which we have identified that natural immunity is there, immunological memory is created. We will use this to create a vaccine. For producing a vaccine, there are going to be two criteria which we will have to look into. First, of course, is the aspect that immune memory should be created naturally through a normal infection process, because we are going to base our vaccine on the normal infection, natural infection process, or you can say immunity to the disease should be there. That is going to be the criteria. Second thing is, okay, let's first talk about the first aspect. We are looking for a disease for which immune memory is created, and it is created for a sufficient time period. That is when it is the point. If there is a disease for which immune memory is for 2 days, that means after 2 days we will have to vaccinate the person again, because the whole concept of memory would be lost. What do we do, or how do we do it? We are giving a vaccine as a preventive measure. Okay, we are giving a vaccine as a preventive measure to make sure that the person does not get the disease. Before getting the disease, we will give the vaccine. That's the concept. So, we are going to exploit this whole idea. इससे पहले कि first attack हो, इससे पहले कि first exposure to a particular antigen is going to take place, ये first exposure हम खुद ही दे देंगे. What we will do is, we will give the antigen to the person. We will give the antigen to the person in the form of a vaccine. B cells के लिए तो ये antigen है. Naturally आए या आप vaccine से दो, B cells do not differentiate between that. So B cells will recognize this antigen, they will produce antibodies, and the most important thing is they will produce an immunological memory. With this memory, once this memory is in place, whenever the antigen comes again, so it means naturally when a person has the chance of being infected, the antigen will come, the body already has a memory which we have created with the help of a vaccine. It will produce so many antibodies that the antigen will be destroyed, the person will not get the disease. That is the idea. Okay. And suppose there is an antigen which has an immune memory of 5 years, so till 5 years the vaccine is good. If there is an antigen which gives this immune memory for the whole lifetime of a person, that means once you are vaccinated, you are immune to that disease for lifelong. So the first criteria, or the first thing which we are looking for, is to have immune memory. And the second thing, and the second important point is, we have to give this antigen in a way as to not cause the disease. आप देखो कि antigen, the purpose of the antigen is to cause the disease. That's why it's there in the body. We don't want that. If I am vaccinating somebody against your, what should I say, suppose I'm vaccinating somebody against polio. And if I give the antigen and the person gets polio, right, after that he might be immune, but that polio has already deformed his leg. So what is the point of it? That's something I did not want. I did not want a deformity. I did not want the person to suffer from the disease. Now I cannot give a vaccine and make the person suffer from the disease. That's entirely stupid, no? So we will not be stupid. The smart thing which we will be doing is, we will be giving an antigen in a way that it does not cause the disease. So what is the optimization point here? आपको एंटीजन भी देना है, आपको इम्यून मेमोरी चाहिए, बट डिजीज नहीं चाहिए. So we have to give the antigen in a form, in a particular form, that it does not cause the disease. At the same time, it still can sufficiently produce an immune memory. To do this, we have different ways. We have devised different methods to be able to do this. Okay. What are those different mechanisms? Let me talk about that. What you need to remember here is that we are exploiting the body's own mechanism in which it produces immune memory to give a high intensity immune response, and for that we give the first exposure of the antigen on by ourselves in the form of a vaccine, which will produce the memory but not the disease. Now what are those different forms of antigen? That is our next point of discussion. Okay, and I will take your questions after that, so just wait. Okay, the first form of vaccines, or we can call this as types of vaccines. The first is live attenuated. So let me take the example of poliovirus. I want to give a person a vaccine for poliovirus. Okay, so what I will do is, I will take the poliovirus and I will make it weak. So I have the potential in the lab, I can weaken a particular virus or a particular pathogen, anything which we want to use. Vaccines we can make for everything, all kinds of pathogens, we can make vaccine against them. So I will take my poliovirus, but I have treated it in the lab to make it weak. It is alive, but it is weak. That's what I have called live attenuated. Why have I gone so far? इसी के नीचे लिखती हूँ. My virus is alive, but it is attenuated. Attenuated means weak. Okay, so I have weakened it. So because it is weak, it will not be able to cause the disease. But because it is alive, their B cells will recognize it, they will produce some antibodies, they will give an immune memory, and the person will be protected against polio. So uske baad, any time after that, when the real antigen, real polio virus comes, your immune memory will be triggered, so many antibodies will be produced that the virus will be destroyed. Okay, but because we have given it in a weaker form, it will not cause the disease. Okay, that's one way in which we can give the antigen. Another way can be inactivated killed vaccine. Now what does this mean? Inactivated killed means now we have killed it, it is dead. Again polio का example लेते हैं. I have polio virus, I am going to kill it. But even after being dead, that dead body of the poliovirus, it can have certain things, okay, like some proteins on its structure, which can still be recognized by the B cells. So some antigens would still structurally be present even if the virus is dead. Okay, so you send this inactivated killed virus in a person's body in the form of a vaccine. Now because it is dead, it cannot cause the disease, right? But the B cells are still able to recognize some structural components. It can be proteins, it can be polysaccharides, it can be anything depending on different pathogens. So even when it is dead, the antigenic component, whatever it was triggering that antigenic response, that is still recognized. Once the B cells recognize them, they will produce antibodies, create a memory, and when the actual attack is happening, they will produce antibodies to counter that antigen. This is the second way. Now, as I have told you that an antigen or antigenic aspect of the B cell can be recognized by that small component. So we have developed this mechanism also. We call it the subunit vaccine. In which whatever is causing that trigger, we study the pathogen and we find out कि अच्छा इसकी जो protein है that is the antigen, इसका जो polysaccharide है that is the antigen, इसका जो lipid layer है that is the antigen. Whatever component we recognize that this is responsible for being recognized, this is what is being identified, that and just that part is picked. आपने एक वायरस लिया, you recognize that okay, its proteins are antigenic. You have a bacteria, you find out कि the polysaccharides present here in the layer of the cell, that is the one which is actually triggering the antigenic response. So you just cut that out और सिर्फ उतना चीज, proteins, polysaccharide or any other component, just pick that out and give it to the person. Now just with the proteins the disease will not be caused. So but क्योंकि वो antigen का part is there, B cells will still recognize them, produce antibodies, produce immune memory. Okay, in this subunit also sometimes what we can do is we can have more than one antigen also. For a particular pathogen, if we recognize there are 2-3 things which are responsible for being antigens, which are recognized by the B cell. So just to make the vaccine more effective, we can combine two antigens, right, and create a vaccine. Same idea as subunit. Okay. And after this we also have nucleic acid vaccines, the DNA vaccines, the RNA vaccines, but for the scope of our discussion here, we are not going to go into that concept because we have not studied DNA and RNA. When we study DNA and RNA, probably in the next class or the class next to that, so that at that point of time we will discuss the nucleic acid vaccines. So for DNA RNA vaccine you have to wait a little bit more, right? Now coming to this question, how do we decide in which way are we going to give the vaccine? Okay, the way to decide that is going to be based on these criterias and it is going to be different for different pathogens. आपने एक pathogen लिया against which you want to create a vaccine. Okay, now you will see and try for different forms. All these possible forms you try for, there is going to be an experimentation which will take place to try in which form immune memory will be properly produced and in which form the disease will not be caused. Not everything will exist in all the forms. Okay, you have to check. हो सकता है for one particular pathogen it does not produce an immune response once it is inactivated or killed. or there can be a pathogen which in the live attenuated form does not reach the stage where it doesn't cause the disease. Even in the most weakest form, it still causes the disease. Or there can be a pathogen which we cannot weaken without killing. So based on these criteria as to in which form it can be delivered as a vaccine, different types are produced. There is no possibility of a comparison which you can do. Okay, so don't start thinking on these tangents that इसमें से अच्छा vaccine कौन सा है, which is more effective. That's not really a comparison which can be made, because the criteria of selection is going to be based on the pathogen. In what form the pathogen will be delivered so that it can produce the immune response, it can generate an immune memory, and also in which it can be given as a vaccine without causing the disease. Okay, so this will determine it. So we cannot exactly compare, and you cannot exactly ask this question कि इस vaccine को इस form में क्यों नहीं बनाया? नहीं बनाया because it might not be possible to deliver it in that form, and of course we are taking this at a very simplistic level. There are many complications in terms of what are the different clinical effects which can take place, how the body will be impacted by it. So it's a very complex science, the process of making vaccines, but basic idea जो है that you have to understand, that you have to keep with you कि we are using the body's natural mechanism to produce antibodies and an immune memory to create vaccines. You might have observed when children are vaccinated, right, at the early stages when a child is born, it's given some vaccination. So with that vaccination, the child is really troubled. He might have fever or some other features. Acha, child ko chodo. Abhi you have been taking the vaccine, so you know there are a lot of symptoms associated with. Those symptoms are because of the immune response that the body is showcasing. आपकी बॉडी में चाहे स्लो एंड लो इंटेंसिटी इम्यून रिस्पॉन्स हो रहा है विद द फर्स्ट एक्सपोजर, बट देयर इज एन इम्यून रिस्पॉन्स, एन एंटीजन हैज कम इनटू योर बॉडी इज रिस्पॉन्डिंग टू इट. सो सम सिम्टम्स, the symptoms which you get, fever, headache, anything, that means your immune system is working. It is sensitive, it is working, and it is countering an antigen which has come. So definitely some symptoms would be there. Okay. In terms of giving vaccines again, it is based on the idea that how long the immune memory is created. For a lot of vaccines, you might be aware that booster doses are needed. For some it will be like, okay, after every 5 years you have to take, for some it would be like after 10 years, for some it would be after 6 months you have to take the vaccine again. What determines that? That determines how long the immune memory would be created. Okay. As far as COVID is concerned, no questions are going to be addressed here, because I told you earlier also about the disease. Nobody knows anything about COVID. Okay. This is my blanket statement and this is the reality of the situation. We have no idea how the vaccine is working. We have no idea if it is working at all. People with vaccination, they are dying. They say that you are supposed to be having a reduced effect of the virus once the vaccine is given. We do not exactly know that. People with vaccination are getting infected again. People with vaccination are dying. We do not know how many doses would be needed. People were vaccinated with 2 doses. Now the government has said ki third dose de do. Vaccine in that sense and its understanding for COVID and every aspect of COVID is a little less. So please don't trouble yourself unkindly with that, and don't trouble us also. That's also intrinsically put in this. But understand the basic mechanism of how vaccines work. That is the important point here, right? I'll make you write all this also. Okay. First, as I make you write, okay. Now if you have any questions, you can ask. All right. Please write down. Vaccination is a process, vaccination is a process in which a person is made resistant to an infection by stimulating the body's own immune response. By stimulating the body's own immune response. When the body encounters, write down further, when the body encounters an antigen, for the first time, a slow and low intensity production of antibodies, a slow and low intensity production of antibodies is done by the B cells. A slow and low intensity production of antibodies is done by the B cells. These cells remain sensitized. And immunological memory is created. Immunological memory is created. On any subsequent attacks, this immune memory is triggered and a fast high intensity response is produced. A fast high intensity response is produced. This is the rationale for vaccination. This is the rationale for vaccination. Okay. Now let's write about the types of vaccines. Give the heading types of vaccines. First, live attenuated. the pathogen is weakened, write down, the pathogen here is weakened so that it does not cause the disease, but retains its capacity to grow. Retains its capacity to grow. Example लिखो अब इसका. The early vaccines which we have, BCG, MMR, they are the live attenuated vaccine. Even your दो बूंद जिंदगी की, the oral polio vaccine, that is also a live attenuated vaccine. The next one, inactivated killed. The pathogen is killed but certain components like proteins can still create an immune response. Examples here, we have in polio, we have one more type of injectable vaccine, that is of this type. Okay. Then your influenza vaccine, और अगर हम कोविड की बात कर रहे हैं तो जो कोवैक्सीन है, India's own indigenous production Covaxin, that is a inactivated killed vaccine. Then after this, subunit vaccines use specific parts from the structure of the pathogen. Use specific parts from the structure of the pathogen. So we have the example of whooping cough vaccine, tetanus vaccine as subunit vaccines. और इसी में conjugate vaccine भी. Conjugate vaccine is combination of more than one antigens. giving a vaccine through combination of more than one antigen. जैसे हमने एक प्रोटीन ले लिया, साथ में एक पॉलीसेक्राइड ले लिया, and both of them together we have mixed and given it in the form of a vaccine. Combination of more than one vaccine. और इसका एग्जाम्पल है न्यूमोकोकल vaccine. Pneumococcal conjugate vaccine. It is an indigenous vaccine produced by India. Pneumococcal conjugate vaccine is a vaccine which is produced in India, created by India. Nucleic acid vaccines we will do later on. Okay. Nucleic acid vaccines we will study later on. We have DNA vaccines here, we have RNA vaccines here, but because we have not studied their structure, so we will do this topic later. इसको छोड़ सकते हैं. You can write examples if you want here. Moderna vaccine, the RNA, mRNA COVID vaccine, COVID-19 vaccine, Pfizer also, Pfizer, Moderna, mRNA vaccine. Now we have a DNA vaccine also for that, but we'll do more details and examples of this when we come to the genetics part. Okay. Now if we have any questions, we can take them before we go to the next topic. Please tell me that you have understood the basic concept as to how a vaccine is working. I would like to know that. Please reply in the response portal to tell me if you've understood this, because this is important fundamentally. If you have understood this concept of vaccination and immune memory. See, when we think of ideas of one particular pathogen having different forms, okay? For example, like hepatitis में we had 5 types, or polio में we have multiple types. Now what happens in that? Sometimes with those different types, the antigen part can remain the same, and sometimes the structure is so different that the antigen becomes different. Okay, so if the antigen or the types in there, if they are different, we will have to produce different vaccines for all those types. That is why hepatitis 5 types, we have 5 different vaccines. We need 5 different vaccines. C के लिए we do not have. Polio also, because we have identified different strains of polio, we have studied them, we have been able to identify what is the antigen in each of them. So we have a vaccine against all of them. We call it the pentavalent vaccine, right? Now COVID का जो example है, उसमें we are talking about the fact that the strain is changing. Okay, the strain is changing. Now when the COVID virus, it changes from one type to the other, alpha, beta, delta, omicron, और भी आगे अभी नए आते रहेंगे शायद, who knows. So in that, when we look at the structure of the virus, there is a change, there is a new strain, a new variety. That means structurally कुछ अलग हो गया. Now, अगर वो structure अलग हो गया, if that structure is different now, we have to look at that structure and see कि उसमें से antigen part change हुआ है या नहीं. Suppose for the COVID virus, we have some proteins on its structure, and for the B cells, that protein part, that one particular protein may be, that is what is an antigen. Okay. If in that change, अगर alpha से beta जाते वक्त of that other variety, if that protein structure remains the same, ठीक है, जैसे कि ये अगर वायरस है, इसके स्ट्रक्चर पे we said there are different proteins. I hope you remember this part. तो अब ये जो एक प्रोटीन है बीच में, right, this one protein, this, if we consider this to be the antigen, then we have to see कि जब म्यूटेट हो रहा है, जब वेराइटी चेंज हो रही है, जब स्ट्रेन चेंज हो रहा है, what the study is needed is this antigen part also changed or some other part has changed? If all of these other proteins change, this antigenic protein, this remains the same, then your vaccine continues to be effective. Okay, but if this antigen, what was being recognized by the B cells, जिसके against B cells ने सीखा है antibody बनाना, the part, the component which was the antigen for us and against which immunological memory is stored for production of antibodies, if that itself has changed, then the vaccine will not be effective. This is the basic idea. If it is the same, if this is same, other parts have changed, right, then the vaccine will also have to be different or it will not be effective. That's the main idea. Okay. Now, any questions? Do we have any questions? B cells produce antibodies, but what is the work of T cells? T cells are memory cells. Kindly repeat the concept of conjugate vaccine. Rest of the concepts are very well understood. Conjugate vaccine कुछ नहीं है, दो antigen है, nothing else. Okay. Conjugate vaccine में we take two antigens, mix them together and give it in the vaccine. Subunit में we take any one component as an antigen and give it. So this is just a way of having two antigens together and giving them as a vaccine. What about T cells? See, T cells also have a similar mechanism. Okay, they will give a response. We said they give a killing response. They also have memory, right? Their system is also working side by side. It's not just the B cells which are attacking the process of a pathogen or an antigen coming in. T cells are also doing similar mechanism, similar idea, but for vaccination, the main concept which we focus on or which we are trying to trigger, that is going to be of B cells. T cells are doing their job of cell mediated immune response, which we have studied. That is being done. Okay, similar thing. They will recognize, they will go attack, destroy the pathogen, the antigen, the unwanted material. Okay, they will also store this in their memory. So they are an important part of the immune system. Ultimately, it's both B cell ka part and T cell ka part. Together they will work along with many other things in the body which are going to be responsible for providing you the holistic immunity which you get. However, the vaccine mechanism, when we study about that, or when we are trying to induce this response from outside, we use the antibody mechanism. That's the only point of difference. Okay, what else? Okay, why something is changing? That is naturally happening. All living organisms in nature, they are going through certain changes. While making copies, while producing new organisms, change can come. It is just that when a virus is based on RNA, the change is easier for it to happen. Okay, और ये concepts हम और पढ़ेंगे जब हम genetics पढ़ेंगे. ठीक है? Okay. Any more questions if there are? we can take them, because I really want this topic to be understood properly. Based on this idea of antigen antibody production, we will divide our adaptive immunity now in two categories. कॉन्जुगेट को क्या डिफाइन करना है? वाई आर यू गेटिंग सो कंफ्यूज्ड विद द कॉन्जुगेट वैक्सीन? सिर्फ सिंपल वर्ड है, देखो कॉन्जुगेट, उसके नीचे लिखो टू एंटीजेंस टुगेदर, हो गया, बस यही है डेफिनेशन, खत्म. ओके, नाउ फॉर प्रोडक्शन ऑफ antibodies, when the body is producing antibodies, we say it is active immunity. The basic mechanism of antigen antibody, your body comes across an antigen, it produces antibodies. The job of antibody is to destroy the antigen. Basic mechanism. Okay. Now this antigen antibody reaction, when it is taking place in the body, we call it active immunity. Further, we can talk about active immunity having two varieties. One is the natural way. Natural way would be like when you have any disease, when you have any infection. You have chickenpox virus in your body starts producing antibodies. Simple mechanism जो हम अभी तक पढ़ रहे हैं. Okay, so when your body is going to be producing antibodies against any foreign body, any infectious agent which has come, that would be natural active immunity. And the second mechanism which we have done is artificially creating this active immunity, and that we are doing through vaccination. So vaccination mein what we are doing is we are giving the antigen from outside. So a person is not being infected on their own, but we are giving an antigen to a person from outside. Okay. That's the whole idea of vaccination, right? Polio के लिए we are giving the person polio virus in the form of a vaccine. So artificially we are giving the antigen so that the body can produce antibodies, and that is the definition of active. Your body produces antibodies, that means it is the active immunity we are talking about. Natural हो सकता है जब खुद से infection हो. If we are giving the vaccine from outside, that is artificial active immunity. Okay, just write this down in one line. Pentavalent. Pentavalent कुछ नहीं है. पोलियो जो है, it has 5 strains. There are 5 strains of this polio virus. जैसे कोविड के बहुत सारे strains आ गए ना, तो polio has 5 strains, and to counter all these 5 strains, we have made one vaccine which is going to work against all these 5 strains. So together we have combined that in one, and we call it the pentavalent vaccine. ठीक है, थोड़ा सा और concentration से सुनना है, right? So that you don't get stuck with every term that is coming in. Write down active immunity. Active immunity, production of antibodies, production of antibodies by the immune system of a person, by the immune system of a person in response to the antigen. In response to the antigen. In response to the antigen. Right? Then you write down natural when there is an infection. Any infection. And second type you will write down artificial active adaptive immunity through vaccination, where you give the antigen from outside. How to understand antigen come from outside or natural infection? Infective मैंने लिख दिया था, infection है. आपको पोलियो हो जाए, ठीक है, खुद से, राइट, नेचुरल इन्फेक्शन हो गया, नेचुरली, राइट. I give you a polio vaccine, in that you get polio virus, live attenuated form of polio virus, 2 बूंद जिंदगी की, right? That is artificial. It is coming from outside. Coming from outside, not a very precise term, okay, but you can say we are artificially giving the antigen even when you were not infected. As a preventive measure we are doing that. That's the whole point. Pentavalent is not a conjugate vaccine. Pentavalent just means 5 vaccines in one vial in one dose. The basis or the mechanism of those vaccines can be anything. So if it is an oral polio vaccine which we are giving, so we can be combining 5 vaccines, but ultimately they are individually they are the live attenuated form only. Chalo. Next is a passive form of immunity. Now, the mechanism is this, antigen or antibody. Okay, your body is supposed to produce antibodies, but because of some reason, sometimes the body is not able to produce the sufficient antibodies. Okay, or maybe it is going to take a little longer time for the antibody production, and by that time the person's body might be severely damaged. So, if we can find a way of giving this antibody from outside, then also we will achieve this. If I know what antigen has attacked the person and I give the antibody from outside, I can prevent the person's disease condition. Passive immunity yahi hai, that we give the antibody from outside. Mechanism will be the same, the antibody will go and attack the antigen, but here the body is not involved in production of antibodies. The examples which I am going to give you here, again, natural, in nature how this happens. We have one example, one exception to this idea, and that is mother's milk. Now when a baby is born, his body is not in a condition to produce all the antibodies. His immune system is weaker, so the baby cannot produce all the antibodies. Okay. So if the baby cannot produce all the antibodies, how is he going to be protected from the diseases? He is going to be protected with the help of mother's milk. The mother's immune system is well developed. She is producing some antibodies and she can directly give those antibodies to her child through mother's milk. So mother's milk, along with all the nutrients it contains, it also contains antibodies for protection of the baby. That is why there is such a significance of breastfeeding. Why? Because without this mother's milk, the baby will not be able to get those antibodies. If you are feeding your baby on cow's milk, so that means he is getting antibodies which the cow is producing. So cow's antibodies will be for the diseases which cow is threatened with and not with the human diseases. So the baby misses out on those antibodies if mother's milk is not there. So naturally bringing antibodies into the baby's body is a passive form of immunity, because the baby's body is not it is not producing antibodies, it is coming from somewhere outside. Okay. Another case in which we can do this passive immunity thing artificially that is if we give antibodies to a person against a particular disease for any reason. Suppose there is a snake bite which has happened. Okay, a person has been bit by a snake and now the snake's poison is an antigen and it is very rapidly acting. So before the body can recognize it and the B cells will produce antibodies, the person is going to die because the response of that antigen is very, very fast. Faster than how B cells or the body can combat it. So what we have an anti-snake venom. Right, an anti-snake venom or antidote to the snake poison. What is that? Basically it is a group of antibodies only, already prepared antibodies which we can give to the person, and immediately that high dose of antibodies will combat my antigen, will combat the snake venom, and the person will be saved. So giving antibodies from outside, artificially prepared, that can be done not just for snake venom but for many different cases. COVID-19 mein we have this thing called plasma therapy. What is this plasma therapy? It is giving antibodies to the person from outside. And why do we need to do it? See, the immunity levels of different people are different. The immune response which different people generate, they are different. So maybe there is a patient who is not in the best of his health and his body is not able to sufficiently produce antibodies to deal with the disease. ठीक है, COVID-19 infection की बात कर रहे हैं. And then there is another person who has recovered, who has healed from COVID-19. So his blood, the person who has been treated, who is healed, his blood will be holding a lot of antibodies. Okay, antibodies against that COVID-19 infection. So what we can do is, we can take antibodies from his plasma. Plasma is the liquid part of the blood. We can take antibodies from there, give it to the other person whose body is not able to sufficiently produce antibodies, and we can save that person. Okay, so for passive immunity, please write down. It is the immunity in which antibodies are not produced in the body, but are given from outside. Okay. And then you can write the types. Vinita has asked, what does passive exactly mean? Passive means the body is not doing the job of making antibodies. That is what active and passive is. अगर body खुद मेहनत करके antibody बना रहा है, right? The body is making the effort, the body is producing antibodies, active. If the body is not making antibodies, still it is receiving antibodies from somewhere. So basic response antigen antibody wala ho jayega, but the body has not done anything. So that is passive form of immunity, because actively you are not involved in production of antibodies. Okay. Now solve one question, then we'll do one last concept of herd immunity and we'll go on from there. Let's see how much you've understood. UPSC previous year's question needs a different kind of understanding. So this question will tell you how much you've understood. What is the difference between poison and venom? This is not a question which I can answer. Sorry. Okay, so the answer which I am getting is D and B. Okay, Vaishali, your question on how do we differentiate between active artificial and passive artificial is the fact कि what are you giving, please focus on that. Active immunity में, vaccination में आप antigen दे रहे हो. You need to go a little deeper into these topics. I had written that on the screen also. आपने भी लिखा होगा जरूर. Active में antibody production is going to take place in the body. Only the antigen you are giving from outside. Passive में you are giving the antibody. It fundamentally changes the whole thing. Okay, just try and see what you have written. B, A, अच्छा C भी है. All options now have been covered. Widespread resistance to malarial parasite to drugs like chloroquine. So they have induced the concept of drug resistance. Chloroquine, which is the drug for killing the plasmodium, the malarial parasite, that is now there. So there are attempts to develop a malarial vaccine. Why is it difficult to develop an effective malarial vaccine? First point that needs your attention here is, the question is, why is it difficult? We are a little used to taking on statements on being correct or not. All these 4 statements can be correct. Okay. There is a possibility in a question like this that all statements can be correct. However, it doesn't matter, because you have to answer this question. What is the reason for it being difficult to develop a vaccine? That's the point here. Vaccine बनाना मुश्किल क्यों है वो बताना है, and not the fact that you are supposed to identify the correct option. ठीक है? Now, how? Let's see all these points one by one. Malaria is caused by several species of Plasmodium. Now we all know that this is correct. We wrote 2 of these species also, Vivax and Falciparum. Okay, but that cannot be a reason why we cannot prepare an effective vaccine. Like just now I told you, polio mein we have 5 varieties and for all 5 we can have vaccine. For hepatitis you have studied 5 types, out of this for 4 types we can produce vaccine. Even if there are different varieties, that doesn't mean that it should be a problem with making vaccines against them. Okay, why? Because this may not be a problem if we know their structure. If we know what are the different types, we can have vaccines against all of them. Okay, so this is not a problem. This statement is correct, absolutely correct, but it's not the reason why it's difficult to make the vaccine. Second point, man does not develop immunity to malaria during natural infection. Third one, vaccines can be developed only against bacteria. So C तो definitely गलत है. Anybody who is has put C has probably put it by mistake, because इसमें तो confuse होना का कोई मतलब ही नहीं है, right? We know that vaccines can be created for other pathogens also. We have been talking about the viral vaccine in such detail. Okay, so C is definitely incorrect and it should not have been anybody's answer. So this question actually is between only 3 options. Okay, ये तो बहुत ही basic हो गया. Last statement, man is only an intermediate host and not the definitive host. Okay, now what does this mean? This means this discussion is about some diseases in which a pathogen goes through multiple hosts. Like in we spoke about a disease called teniasis, or chalo teniasis छोड़ो, swine flu. It happens in pigs, then it happens in man. Okay, so more than one host for the pathogen. But malaria is a vector-borne disease and you have studied the malarial cycle. There is no other host. There is only a mosquito which is a vector. Mosquito के लिए we have done this. Mosquito is only supposed to be the carrier. It is not supposed to be having anything to do with the disease. We established that. It has nothing to do with the disease. The mosquito does not get the disease. It is not a host. Host is somebody who will get the disease. Man is the definitive host. For malaria, there is only one host. We have studied the cycle of malaria. So man is the definitive host for malaria. और कोई organism नहीं है. जैसे anthrax है, बीच में cows are there, or things like that. You know, bird flu में birds are there. But here there is nobody else. Man is the definitive host of malaria. तो ये तो statement ही गलत है. We are left only with these 2 statements are correct. The first are incorrect. वो statement ही गलत है. The first statement is correct, but it's not the answer. So the answer is B. Man does not develop immunity to malaria during natural infection. So people keep getting malaria year after year. That means a long-term immunity for malaria, or the immune memory for malaria, is not very long. And because of that problem, we are not able to have an effective vaccine for malaria. Okay, if we have to give the vaccine again and again at short intervals, it doesn't really, we cannot really say that why we are, this is an effective malarial vaccine. So naturally जिसकी immune memory नहीं बन रही है और natural immunity we do not have against that disease, then we will not be able to extract. But a disease like chickenpox, very effective vaccine we can produce. Once you give the vaccine, you will have very, very good results for lifelong. Okay. Now see, slight variation in immunity or how long the memory will be there, that will depend on the pathogen. That is the property of how the pathogen and the mechanism of its processing is taking place in the body. For some people who are immunocompromised, who have some diseases linked with their immune system, for them in a normal kind of disease also they might not have immunity. Like Chicken pox can happen to some people again, twice in their lifetime. If that is the case, that means these people have lower immunity. That is why that lifelong protection is not there, but for most people it would be there. Plasma therapy is artificial passive or natural passive. No, we can see अब ये थोड़ा सा technicality है Sayush, but we will be considering this to be artificial passive. Okay, I understand the fact that the antibodies are not produced in the lab, they are naturally derived from a person. So maybe plasma therapy doesn't exactly fit in that category. So passive at least surely we can say, but because we are giving it from ourselves, we are administrating it, so we will use the term artificial passive. ठीक है? Now this is a very important concept. We keep hearing about this. This will also give you a little bit more idea as to why we are talking about COVID-19 vaccination when it is not long term. How gender biased options are. So I don't get this comment at all. Let's talk about herd immunity, very important concept. This means when a sufficient amount of population is immune to a particular disease. Once we have achieved that situation, we say it is herd immunity. What do we mean by this? This diagram is very, very important to visualize this concept. Okay. Now, the blue people here, they are the people who are protected from a disease. Okay. Now, what happens? They can be protected through a vaccine or through the fact that they have already been affected. Irrespective of that, but people who have a sufficient amount of immunity or immune memory in place or cannot get a particular disease, they will be called protected. Those who are not protected, they are the orange ones. They are the at-risk people. And there is one person here in between, you can see this white person, which is going to be the contagious person who will be spreading the disease. When we talk about the herd immunity, okay, we are going to say that when 70 to 80%, the figures will vary at different texts when you will be reading it. Concept बस आपको समझना है. 70%, 80%, what we are trying to say is majority of the population, when it is protected from a particular disease, that condition is called herd immunity. What is the advantage of this? अगर 70 से 80% लोग in a particular population, if they are protected against a disease, this contagious person, when he is out in the population, the chances of him meeting the at-risk people is less. Because most of your people, majority, 80% of your people are protected. आप इसको color coding से देखो. The white person, because most of the people are blue, the white person's chance of meeting an orange person, the at-risk person, is very, very low. The probability is low. Okay. Because this probability is low, the chances of this disease spreading will be less. If this contagious person is not able to meet or come in contact with these at-risk people, the disease progression will stop. And that is what we want to achieve in herd immunity. आप अपने जो blue people हैं, the protected people हैं, you increase their number so much, 70 to 80%, that they will not be able to come in contact with the infected person or with the contagious person. ठीक है? Think about COVID-19 here. As of now, we know this for a fact that COVID-19 infection, when anybody gets COVID, so we said for 2-3 months after that, that person is protected. Okay, so there in this diagram only imagine that there are a few people who have just had COVID and for the next 2-3 months they are going to be safe. Now this is one group of safe people. The other people we are trying to save through vaccination. Some other people we will vaccinate so that at one point of time they will all be, or the majority people or 80% of a population, they would be protected. Of course we know that protection will go away after 4 months, but वो एक time period हमने ऐसा create करना है. The idea is that at one point of time, majority of the people in a population should be immune, should be protected for a disease, right? They might lose their immunity after a point of time, but by that time you will have stopped the progression of the disease. That's the idea, that's the point, that is the evidence why we keep talking about achieving herd immunity. That after that point or at that point, when, because this person also, there is this one contagious person in this population. So after some time, 15 days, 20 days, whatever is the scope of the disease, this person will also be free of the infection, right? So the disease itself, the disease progression will stop. Even if people lose their immunity after 5 months or 6 months, that will not make a difference because now the disease is not there. And this person, because the population on the whole is being protected, will not be able to come in contact with the at-risk person. That is our main idea. So diseases will die once herd immunity is being achieved, because they will not be able to spread. The chances of the infected person meeting the at-risk person is low. Okay, this is the basic idea. So you can just write down herd immunity and you can write this: when enough people in a population have immunity in a way that the disease cannot spread. Very important concept, always in news. The numbers can vary, but it's usually 70 to 80%. इतना population होना चाहिए to stop or the cutting the spread, the community spread which they talk about, cutting that spread, cutting that line of transmission. What is the difference between strain, mutant and variant? There is no difference. These are interchangeable terms for the level of understanding which you need. They mean the same. Aniket. Now we are finished with our topic on immunity. We are going to start talking about different systems in the body. I hope you've understood the concept of immunity. The different terms should be clear in your mind. What is innate immunity? What is active immunity? All that. Please try and revise it today. I'll tell you a chapter also where you can get some insights if you want to go into further reading, right? But it will give you a lot of personal understanding about what's going around in the world right now. Remdesivir is a medicine. Yes, it is a medicine, it is a drug. It is an antiviral medicine. It has absolutely nothing to do with immunity. just like antibiotics I explained. So the first system which we are going to do is the digestive system. Of course, we do not need a lot of technical details here because the kind of questions which are being asked, they are kind of basic. So we'll take an overview of the different systems. We'll only focus on aspects which you need to know, right? There is no notes, no dictation which is going to take place here. You're supposed to take running notes. All of it is actually going to be on the board, so I don't think you will face a problem. Let's start with the digestive system. When we eat food, when we take nutrients, they cannot as such be used in the body. They have to be broken down to be used. So if there is a carbohydrate, it has to be broken down into its component. If it is a fat, it has to be broken down into its fatty acid. If it is a protein, it has to be broken down into its amino acids. Only then the body will be able to use it. So digestive system is to break down food into simpler forms. Breaking down of food into simpler forms is what is digestion, and that is what the digestive system is for. For doing things, different parts of the body produces different enzymes. For breaking down different components of food, we have different enzymes being released in different parts of the body. So in which part which enzyme is being released, that's what we have to learn. We will start with the mouth. In the mouth we have salivary glands which produce saliva. And this saliva is going to break down the carbohydrates with the help of an enzyme called amylase. We have the amylase enzyme, and this amylase enzyme is going to do the job of breaking down carbohydrates. So as the food enters your mouth, the first thing which it acts on, or the first thing which it breaks down with the help of saliva, is carbohydrates. Okay. After this, after the carbohydrates have been broken, the food will go through the food pipe. This is the esophagus. This is the, it's also called the food pipe. Esophagus or food pipe, no digestion is taking place here, it's only a channel. So from the mouth to the stomach, food is going to be transported with the help of the food pipe. It's just a connecting channel, no digestion is taking place here. In the stomach now, what we will have, the food is reached the stomach, this is the stomach. We have HCl and we have protease. So protease, by its name you should know, it is going to be the enzyme which will break down proteins. Enzyme to break down protein is found in the stomach called protease. So first carbohydrates in the mouth, then the food comes to the stomach, it will have, or it will be breaking down proteins. Then from there, from the stomach, the food will go to the intestine part and it reaches the small intestine. Small intestine is actually the longest part of your digestive system. Why it is called small is because of its width. It is very thin, its thickness is less. Stomach से food has now come to the small intestine. Small intestine has a number of enzymes. Number of enzymes are produced in the small intestine. We have peptidase for proteins. See, all these nutrients, they are complex structures, so multiple enzymes will be needed. And different enzymes would be acting on different types of nutrients also. So that's why we have more than one enzyme in this digestive system. So you have peptidase, sucrase, maltase here. That means basically it is doing the job of breaking down carbohydrates. So two things which are going to act on your proteins proteins will be broken down by the small intestine and the stomach, and carbohydrates के लिए अभी तक we have done the saliva and the small intestine. Okay. Along with these basic places or the main places from which food is passing, we have two glands also which are going to help. Okay, we have two glands which are going to help. One is liver and the second one is pancreas. Now the food is not going to go into these glands, but they are going to be producing some liquids and they will be sending them to the small intestine. Okay, what is the liver doing? Now liver as such does not have any specific digesting enzymes, but it has a very important role in fat digestion. Liver produces bile, and bile, though it does not contain any fat digesting enzymes, still it is very important for helps in fat digestion. Bile helps in fat digestion without having any enzymes. So if it doesn't have fat digesting enzyme, what does it do? So think of fats as very large and tangled big globules in a way that their structure is so complex, so entangled that it is difficult for enzymes to act on them. So the job of bile here is to untangle them, to free them, or to make those from those large globules, it will make smaller globules, smaller, simpler structures, so that they can be broken down by the enzymes. Okay, so remember this, liver produces bile. Bile does not contain fat digesting enzymes, but it helps in fat digestion. I am hoping you are writing all this down. Now, once bile is produced, it is stored in an organ called gallbladder. So gallbladder is not having any production of anything on its own, but what it is only doing is that it is doing the job of storing bile. The storage of bile is happening in the gallbladder. So liver produces bile, it is stored in the gallbladder, and then it is used to be released into the small intestine. So small intestine में अपना production तो है ही, तो this can be called intestinal juice. Along with that, it is also receiving bile, right? and another gland called pancreas. It can be said to have pancreatic juice. Pancreas is also producing a juice, is also producing a liquid which it is releasing into the small intestine. What kind of enzymes it has? It has enzymes for all three nutrients, proteins, lipase for your fats and amylase for the carbohydrates. So pancreatic juice contains protease, lipase and amylase. Okay. Now we are in the small intestine with three fluids, three juices. You have pancreatic juice, you have intestinal juice and you have bile juice coming from the liver. With all these acting on the food is completely digested, completely broken down into its components ready for the body to use. How will the body use all this is that small intestine say absorption of nutrients takes place and the nutrients goes into the blood. Everything which the body has to take from one place to the other, it will be taken with the help of blood. So, in small intestine now everything is broken down, all the nutrients which the body needs, they will be sent into the blood in the small intestine. Okay. But not everything which you eat is useful. Okay, some things the body will not be able to break down, not be able to digest, it will be extra. So all those extra things, they then go into the large intestine, this part. In this large intestine, now water is also there along with food. So the water is absorbed in the large intestine. The body needs water also. So in the large intestine, absorption of water takes place. In the small intestine, absorption of nutrients takes place. And after water is absorbed, whatever is left, whatever is undigested, whatever is the waste, that has to be removed from the body and it will go outside the body through the external opening called anus in the form of your stool. So your stool or your feces is basically the undigested component of food. Whatever the body is not able to digest, whatever the body is not able to process, that will come out from the external opening of your digestive system, that is the anus. Okay, let's just go through this once again. I'm hoping you've written all this down. Food comes into the mouth. First thing to act on food is saliva. Saliva contains an enzyme amylase which breaks down carbohydrates. After this, the food passes through the food pipe. It is called the esophagus, reaches the stomach. You have sphincters being mentioned here. Sphincter basically is a muscle band. So stomach, you can see its position is slightly tilted. So once digestion is to take place here, we need to keep the openings shut. ठीक है, ऐसे नहीं होना चाहिए कि जैसे acid is being produced. If the stomach remains open, this acid will go into the mouth, which sometimes happens if we are not sitting in a proper position or if there is something wrong with this sphincter. So we have that acid reflux. Okay. But in normal conditions, it is important that the stomach remains sealed when digestion is taking place. So for that, it has muscle bands. Okay, stomach में क्या हो रहा है? We have an acid being produced and we have an enzyme called protease which is going to now break down proteins. Okay, after this food goes to the small intestine less in thickness but greater in length, longest part of the digestive system. Small intestine produces intestinal juice which has enzymes peptidase, sucrase, maltase. So that means carbohydrates and proteins it is going to break down. Small intestine is getting two other glands secretions. One is the liver. Liver produces bile. Bile does not have any fat digesting enzymes but still helps in digestion of fats. This is all that you need to know. And then pancreas. Pancreas produces pancreatic juice. Pancreatic juice में you have protease, lipase, amylase, breaking down protein, fat and carbohydrate. So both these fluids, they are also coming into the small intestine and they are completing the process of digestion. All your food is now broken down into smaller, simpler parts, and with this what is also happening is that there is going to be absorption of nutrients into the blood. So if you have a question, where is food being absorbed? Where are the nutrients being absorbed? The site for that is small intestine. And whatever the body does not want, cannot digest, cannot break down, not needed, it goes to the large intestine. All the excess water is absorbed here. Absorption again in the blood, so that blood can take it to other places. And with this, what's going to happen is whatever is still left after water is being removed, that will be thrown out of the body through your external opening, the anus. Amylase is for. Why do you even ask such questions? Can't you see? देखो लिखा है. ऐसे ऊपर भी देखते रहना है. सिर्फ all senses you have to use if you have to sit in the class. Eyes, ears, and of course the brain. Or is my handwriting so bad? I mean, I know I'm not very good, but I think you should be able to make out carbohydrates. Herd immunity का definition so many of you are asking. I did not really make you write a definition because it was here. Slides. ये भी use करनी है. They are given to you for a purpose. इस पे लिखा है देखो. When enough people in a population have immunity that the disease can't spread. यही है. इतना ही है herd immunity का definition. Okay. So, eyes, ears, brain. Sanjana, Sangeeta, any specific queries which we have? I am hoping you know they are your coordinators, because in one of the batches what happened was, like the faculty is teaching, so they'll usually ask or talk because we are in a virtual interspace, so they'll take the name of the batch coordinator. Like I've asked, okay, Sanjana or Sangeeta, if there are any queries which we have. Okay, so there was one student, he wrote a very emotional mail to us saying that why are you only talking to 2 children in the class? And then it becomes, in a way, he actually said that I have also paid the fees, why are you having this kind of nepotism, why do you come to the class and say good morning only to 2 people? Okay, so it was actually, we did feel bad for the student that how things he's interpreting, and he was feeling that way for many days before it was made clear that no, they are the batch coordinators. They are the ones who are supposed to transfer the queries. That's why the faculty uses their name, and they are not my favorite students or anybody's. So. Do enzymes can be proteins, carbohydrates and fats? I do not, this is a question from Archie, I do not exactly get this question, but the sense which I can make from here is that, actually I cannot make any sense, but I will still tell you a little bit. Enzymes are proteins. If you remember, we studied enzymes as examples of globular proteins. So all your enzymes are going to be proteins, and for the purpose of the topic which we are discussing, digestive enzymes can be there, or we have digestive enzymes to break down carbohydrates, fats and proteins. And there are enzymes in the body for many other different functions. But if chemically you are talking about their nature, they are going to be proteins. Okay. Sphincter. Sphincter are muscle bands which are supposed to close the stomach. Keep the stomach closed while digestion is taking place. Okay. At both the ends of the stomach, because if the stomach is not closed, then the fluids from the stomach, because it is acidic, we have to be extra careful. They can flow to the small intestine, or they can go back into the esophagus. So we do not want acid to go anywhere else. So we have muscle bands to keep the stomach closed. Okay, that is what is sphincter. If this sphincter does not close properly, we can have problems. The basic idea of acidity which we feel or experience, that is when stuff from the stomach is going back into your food pipe, and you can sense it somewhere around your throat. Okay? Okay. Now let me tell you about some chapters and what we will do in our next class. Next class mein we will do other different systems, starting with respiratory system and other systems after that, and we will most likely be able to start with genetics in the next class. अभी NCERT पे आते हैं आपकी. So you can take your class 7th NCERT, read this chapter number 2, which is on digestion. Then we can do this one chapter which is from your class 12th book, chapter number 8. This is a chapter on health. So you'll have different aspects of health here. You also have, I'm not sure if I told you this last time or not, but still it talks about immunity, different types of immunity definitions. So if you want to read, it will be a little bit more advanced in terms of health and spread of diseases and all those things. So you can selectively see what makes sense to you, and you can use this as a reference. Okay, different ideas of innate immunity and different types, active, passive, all of that is going to be available there. Okay. Please define HCl, ma'am. Okay, I'm sorry, it's my bad. I should not have assumed that that could be a problem. HCl, thank you Ashmita for helping me, is hydrochloric acid. इसके आगे का definition मुझे भी नहीं आता HCl का. ठीक है? Okay. So we'll end the class here. Thank you Sangeeta and Sanjana, and thank you all of you. We'll meet in the next class, whenever that is.